I'm going to talk about ancestry and using diverse ancestry data in genomics research. This is not going to be kind of a puppies and rainbows kind of talk. The conclusion is going to be that this is very hard and we haven't solved these problems, but also it's really important that we do solve these problems. So I'm going to start off just by giving some context to talk about the ethical considerations. This was mentioned by one of the discussants today. So genomics research generally, not just social science genetics, has focused just overwhelmingly on European ancestry samples. And so, this figure here, the red here is the sample size of genetic studies that have been done in European ancestry samples. And the bar over here is the fraction of the population that corresponds to European ancestry samples. So you see that these studies that have been done are just overwhelmingly done in this subgroup of the population that represents only a small fraction of the global population. So why is this bad? Well, if we're only studying a certain subgroup of the population, then the types of policies that we're going to develop, we'll only learn about that subgroup. And so as we develop policies that, are maybe meant to improve welfare of society, those benefits are largely just going to land on this subgroup that we're publishing. And as a result, Alicia Martin published a paper where she says that genomics research, as it's done today, may contribute to increasing inequality and increasing health disparities. So this is a big problem. If the work that we're doing is actually just making the world a worse place, assuming we don't like health disparities. As a result, I think there's just this urgent need for us to develop and use methods that allow us to use data in diverse ancestry samples, if that's possible. On the other hand, if we develop tools that allow us to study genetics in diverse populations, those tools can also be used by bad actors or maybe naive people who don't understand what they're doing. Furthermore, if we don't do this research carefully, did this turn off? Can you hear me anyways? Okay, great. If we don't do this research carefully and so we just do sloppy work in an effort to say like, oh, let's be inclusive, let's use everyone, these sloppy practices can then bleed into the research that's done that may that may badly affect disadvantaged communities. So for example, I saw this paper last week, actually, where they found a genetic variant that increases the risk of mortality of dying from COVID by double. It was some really huge effect size. They find that this genetic variant is found in 60% of people with South Asian ancestries and only 15% of people with European ancestries, and say, oh, this maybe explains some of the differences in mortalities between these different groups. And so, they use this to say maybe this should help us to motivate vaccinations, and they have all these happy conclusions about, isn't this great that we found these differences? However, using the same reasoning, there's this paper that also came out several years ago where they say that differences in the polygenic scores between Jews and Lutherans are explaining the cognitive performance differences between these groups. I think that both of these papers or both of these conclusions are garbage. But we see this kind of research and it may motivate this kind of research that we perhaps don't want to promote. And so, as we're working on trying to incorporate diverse data into the work that we do, I think that we just need to be very careful about the limitations of what we can say, make sure that we're doing it very cleanly, and things like that. So that's just kind of an intro, motivation, things that I think we should keep in mind as we're using diverse ancestry data. I'm going to talk a little bit about population stratification next, because this is the primary complication in incorporating genetic data in diverse ancestry samples in the work we do. So this may be a bit of a review for everyone, but the simplification is helpful for me, at least, when I'm thinking about how we might solve some of the problems of using diverse ancestry data. And so let's imagine just a super simple population. We have a group of people, and then maybe there's an earthquake, and it creates a big chasm in the middle of this population. All of a sudden, people can't get across this chasm. And so we have now two separate populations. Now, at time equals 0, assuming that it was random where this chasm was built, the allele frequencies in these two groups are going to be approximately the same. There might be small differences due to finite samples, but more or less these populations are going to have equal allele frequencies. But over time, allele frequencies drift just by chance, by what genes happen to be passed on from one generation to the next generation. And so if these populations can't get over the chasm, then the allele frequency in this population might go up and this one might go down. And so over time, we're going to see differences like this across the genome, across all SNPs. And so the problem with this now is that this SNP here is predictive of which side of the chasm that you're on. But maybe there's other environmental things that are different between, right chasm and left chasm that have nothing to do with genetics. But if we did a genetic study, and in the superpopulation, this SNP is just going to capture the environmental stuff. So that's the simple example. The real world isn't a series of chasms. And so here's again, it's a bit of a simplification, but I think a useful simplification for how the world is a bit more. So imagine that we have like a model where people live on the unit interval, and so where they are in the unit interval is geographically where they live. And mating is, people are more likely to mate with people who are close to them. So sometimes you call this the cline. So this kind of smooth variation geographically. And people are more likely to mate with people who are close to each other geographically on the cline. So again, you'll see frequencies moving up and down by chance, just based on who's mating with whom, but because these individuals, a person on near 0 is unlikely to mate with a person from 1, you may start seeing differences in allele frequencies across the cline. And so, it's not going to be, it will in general be continuous in this very simple model, but there's going to be some sort of continuous relationship. And we have the same kind of problem. So if we did a GWAS in this population, if there are environmental differences that affect this group differently than this group, a person's genes predict where they are in the cline, and so that's going to introduce bias. Even when we have kind of data that's kind of a smear of ancestry, which is, I think, a bit more realistic in what we're seeing today, because people do mix somewhat based on geography, for example, but maybe also cultural barriers that exist between groups and things like that. So this is maybe the world we should imagine. Yes?
Can you just say what is generating the drift in allele frequency?
So it's just pure well, okay, there's two things that it could be. What I've been talking about here is just pure randomness. And so, if you have like 100 people and they each pass on their genes to the next generation, which gene, which chromosome they pass is a 50-50 chance. And so it may be that certain variants are passed on to the next generation, just by chance, slightly more likely. So there's sort of...
If the population are large, I wouldn't see drift, would I?
If the population were infinite, then yes, you wouldn't see very much drift. But populations are not generally large enough. So these drift things happen over hundreds of generations. And so from one generation to the next, drift is small, but if you look at 300 generations, it can aggregate to something kind of big. Does that answer your question?
Especially if they remain separated.
Especially if they're totally separated during this time.
Wouldn't it be a more plausible explanation or story that the separation is a difference in environment that puts in place different selection processes?
Yeah, and so that's the other alternative. So it could just be that there's selection. It may be that over here it's advantageous to be tall so you can reach the food at the tops of the trees, and over here it might be advantageous to be small so that you can climb into the caves to escape predators. I don't know, something like that. And so it could also be selection differences, but even if there's no selection, you do end up seeing patterns like this over time. And so this is a big debate of how much of the differences in allele frequencies across populations is driven by selection versus drift, and this is like a lot of what Aldo was presenting. This is why the selection question is very hard, but potentially very interesting too, because there's these two factors that may create differences, and it'd be nice to know which one it is, just scientifically. But drift is sufficient to produce this, is the main point. And so even if there is drift, you may find kind of false signals just due to the fact that people from over here are going to have, just by chance, different alleles than the people over here. Great. Okay, so here's some equations, and so I'm going to go back to this linear model to make this a little bit more concrete. So we have some phenotype y, we have a vector of genotypes x, 0, 1, 2s, and I'm going to say that there's some sort of ancestry-related intercept, and so this will capture sort of the environmental differences that a person faces, and these might be correlated with the genotypes a person has. And so if you imagine a GWAS, what we're doing is we're regressing y onto each of these x's, but mu is unobserved. And so if mu is correlated with a particular x, then there's going to be omitted variable bias. And so that's how stratification can kind of creep into GWAS studies. So how do we deal with this? Well, as you saw in a lot of these talks that were done today, the first thing that we often do is we restrict to European ancestry samples. And the goal of doing something like that is that maybe we can find a group such that the genotypes are independent of the environment. So if the sample has homogeneous ancestry, then maybe the expected value of mu is the same for everybody. Mu is maybe constant. Or perhaps the expected value of x is constant across the population, and so that breaks the correlation between x and mu. Hopefully, that's the goal at least. Alternatively, we can find controls, and so Dan just gave this big lecture about trying to find controls which make it so that your genotypes are as good as randomly assigned, and so if you can find genotypes such that x is uncorrelated with mu, given your controls, that also will remove some of the biases from stratification. So those controls could be parental genotypes, maybe family fixed effects, although Dan says that you shouldn't use those. I mean, I agree. But in the reason you do it is because you want to break this correlation. Another thing that's really commonly done is to control for the principal components. And the goal of doing something like, the goal of doing something like that is, imagine, I'm going back to this figure again. So now this is the allele frequency figure where we see that there is, a relationship between the location that a person is and the allele frequency in their population. And then I've also plotted here a relationship between the expected value of this unobserved, intercept effect and the location as well, right? And so if we did no controls, you can see these are both upward sloping, they're correlated with each other, and that's why there's bias. If we control for principal components, what effectively we're trying to do is we're saying, well, let's try to fit a line to the allele frequencies, let's fit a line through these location things, and now all we need is for the deviation from this line, because these PCs are just meant to be like a linear fit of, hopefully capturing a person's location in this very simple example. And so the hope is that the deviations from this line here are uncorrelated across these true groups. Now, in a European sample, what we're essentially doing is we're just looking at a narrow strip. We're only looking at people like who are very close to each other in the cline. And because things tend to be locally linear, if we choose a narrow enough portion, then drawing a line is going to be effective at breaking the correlation because it's going to capture. However, if we have very diverse data, meaning like data that spans the whole cline, then all of a sudden you can kind of see why maybe PCs aren't going to do their job, right? These are potentially very complicated nonlinear relationships, but you need the residuals to be uncorrelated with each other, and there's no guarantee that that's going to be the case. Yep.
If we know we have different populations and fit a PC within population, would that solve the problem?
So there's a handful of problems with trying to separate out the populations. One is that you have to define what a population means too, and the world is pretty fuzzy. We've been talking all day as if there's this thing called the European ancestry population, and in practice that doesn't exist. And ancestry is this continuum. We all kind of have ancestries from different places. I think that theoretically, I'm going to say sure. In fact, I have a project that I'm working on right now that essentially tries to do that, where we try to identify, subpopulations that have similar ancestries and fit PCs within them, and hopefully that helps. But this is open research. I think you would do better doing that than you would just using one PC for an incredibly diverse data set. Okay, so let's talk about polygenic indexes. And so I'm going to extend this linear model just a little bit different from what I showed you earlier, just to make a handful of other points. Specifically, I'm going to make these effects here be individual specific. So instead of them being like the average effect in the sample that we're using, this is actually the true effect for this particular individual. So if for that individual we adjusted we edited their genotype at conception, what would be the effect on their phenotype, and it could be different for everyone. So when we're doing something like a GWAS, we're getting the average B sub ij in our sample, but you could imagine there's some heterogeneity in the B for every person. And then when we're making a polygenic index, it's just a weighted combination of the genotypes. But it's also, I'm going to make another distinction here. J is like all genotypes in the genome. When we make a polygenic index, we only have some set of genotype SNPs that we're using. So it might be that we don't include rare SNPs, or they're just things that aren't included on the chip that we're using. And so we're using a subset of genotypes when we're making polygenic indexes, and that's going to have implications in diverse ancestry analysis. And so where do these weights come from? Well, it's theoretically what they should be is they're the weights that minimize kind of the mean squared error of the polygenic index and the phenotype in our sample. Like this is the thing that we're aiming for. In practice, we try to approximate this with the data that we have, but I think this is maybe just helpful for understanding what the theoretical ideal is. Okay? So that's a PGI. So I'm not going to go through all of these probably very carefully because there's not much time. So PGIs have a handful of additional complications. And so, first, they're not generally comparable across ancestry groups. This is related to the stratification problem. Another problem is that when you make a PGI based in a European ancestry sample, for example, so you do a GWAS in a European ancestry sample, and then you try to predict some phenotype in an East Asian ancestry sample, it tends to not do as well. Just empirically, this is what we see. And then there's another issue of admixture, which I'll hopefully have time to talk about. Admixture is a mess. It's just difficult. So I'm going to go through each of these points just briefly. So here's a figure that came out 4 years ago that I thought was really striking. And so this is a paper by Alicia Martin. What she did is she created a polygenic index for height based on a European ancestry sample. And then she took the 1,000 genomes data and she plotted the distribution of the polygenic index for height in a handful of different ancestry populations. And so you see here on the right in the blue, this is the distribution of the PGI for European ancestry samples for height. In orange here, this is South Asian ancestry people. And then this like dark red here, the African ancestry sample. All right, so looking at this, it is the case that the South Asian ancestry population is a little shorter than your European ancestry populations, but the African ancestry populations are roughly the same height as the European ancestry populations. However, looking at this figure, the mean polygenic index for the African ancestry people is 4 standard deviations below the mean in the European populations. And this is not reflected in like the real, if you measure the height of these individuals. And this is just driven by population stratification, chance changes across these different groups. And so like these kinds of figures are what makes me a little nervous, for example, like of the figure that Aldo made looking at the polygenic score distributions for these different subpopulations. Because if you just did like a random PGI and then looked at a bunch of different ancestry groups, these differences aren't signable. Like the order here, it just happens to be that the European one is at the top, which happens to match the ancestry of the GWAS sample, but this isn't generally the case. Like the order of the mean of these distributions has an unpredictable sign. And so it's, I think there's been a lot of work in the past where they try to make comparisons between different groups of the PGI distributions. And what this paper is claiming is that these differences are generally meaningless unless you do a lot of additional work. Just the raw comparisons aren't very meaningful. And so, so that's that point there. So, how do we deal with that? Well, the way you deal with the biases here are the same as what I talked about before. So if you control for parental PGIs or have family fixed effects or restrict to samples with homogeneous ancestries, like hopefully that solves some of these biases from stratification. The next point is just about the differences in predictive power, and so this is another. Alicia Martin has done a lot of this descriptive work, which is great. So she created polygenic indexes in European ancestry samples, and then tried to predict those phenotypes for a bunch of different traits in Hispanic Latino samples, South Asian, East Asian, and African ancestry samples, and you see that there are kind of systematic decreases in predictive power of these polygenic indexes in these different groups. So like African ancestry individuals, on average, have a predictive power, the polygenic index in African ancestry samples have a predictive power that's like a quarter of what you see in European ancestry samples. This is not anything to do with European or African ancestry people in particular. If we had done the GWAS in the African samples and then predicted in these four groups, it would just go in the other direction. So Europeans would be down here in that case. It's just a function of the mismatch of the populations. Just very briefly, there's two big reasons why. I'm not going to talk in detail about this, but one is, you can kind of see in this model, one big difference is that the Bs may be different between these two different groups. And so if the Bs are different, then the weights that you're using in the European ancestry samples are just the wrong ones because the effect of each SNP, the average effect of each SNP is different in the two groups. And the other is that the correlation structure is different between these two groups. And this goes back to the ungenotyped SNPs. So, imagine that there are 3 SNPs, and these two are correlated with each other in the European ancestry sample, and these two are correlated with each other in the African ancestry sample, but you don't see this middle one. Then your PGI is going to try to load the information about middle SNP onto left SNP. Or is it? Yeah, left for you guys. Whereas if you then try to create a PGI in the African sample, this SNP doesn't say anything about this SNP in the African ancestry sample, and so you're going to lose information. It's just not going to be optimal. So how does this limit research? Well, if you do the controls for stratification, like technically you might have some predictive power. You maybe could say something about differences in risk between different groups based on their PGIs. However, this leads to huge losses in statistical power because we're needing to restrict first to our small homogeneous samples, and then we have even lower predictive power in these PGIs. And so, it's if you're doing analysis where you're saying, well, let's compare the differences in predictive power between groups, you can't be sure if that's because there's differences in like heritability or interesting stuff, or if it's just due to the fact that you happen to have slightly different ancestry compositions in these different groups. So what can we do? We just need more data, and this is something that's underway. We need more data in non-European ancestry samples because that will increase the predictive power in those other groups. There are methods that are being worked on that directly account for the differences in LD, which is the correlation structure, and the SNP effects. So there's like MAMA, which is something I'm doing, or PureSCX, which just creates polygenic scores that use the European ancestry data to say, well, how predictive can we maximally get in some other population? I'm going to skip this other thing. And then I also just put this point here, like a lot of this problem is because we're relying heavily on these PGI weights. And I know Jason Fletcher, for example, has been talking a lot lately about, well, maybe we should think about methods that don't use PGIs and that just use the molecular level data. Maybe there's some way forward there. I don't have time to talk about admixture, but maybe just explain why it's hard. So admixture is this idea that, we try to slice and dice our data, and say these are the Europeans and these are the Africans and these are the East Asians and things like that. But in reality, a lot of we're all mixtures of a bunch of different ancestries, and this is especially true in certain populations, like African American population. They have ancestors from the African continent and from the European continent in different proportions, and so this makes the whole idea of what ancestry is incredibly complicated. Because you look at a person's, this is like an example of an African American genome, and the blue parts of their genome are African ancestry, and the red or I think blue in this case is European, and red is African ancestry. And so you can see that where their SNPs are come from different places. So a person is diverse even within their own genome. So not even diverse across people, but diverse within a person. It's hard to think of how to solve this problem. So how do you, by definition? But I don't know what happened to this slide. but like some of the issues, how do you define a homogeneous population if you have an admixed population, right? People are already diverse, just, a single person has diverse ancestry within themselves. So, how do you deal with that? The correlation structure is complicated when you have admixed populations because it depends on, which ancestry the particular SNP you're looking at came from. It's hard. And there's not really solutions. So I'm saying there's no established approaches. You can try controlling for the parental PGI. That's always great. Maybe restrict to people with similar admixture profiles. Maybe PCs. It's not clear that works. Like, everything's a disaster when you have admixed people in your data. So in conclusion, it really is important. So I've been saying, like, it's all hard, there's no solutions, but it's important for us to figure this out because the questions that we're interested in as economists, hopefully are helpful and applicable to the entire population, and we can't currently study the whole population at once. And so, how are we going to get over this? Well, family-based methods are great. If we can't do family-based methods, I think the next best thing is to maybe split your data into homogeneous samples and then meta-analyze when you're done. And then also just, I say waiting, this is a super active area of research. There's tons of money being pumped into data collection and methods development and things like that. So I think within the next few years, maybe this talk could be more uplifting. But we're learning. So that's everything. Yes.
So a lot of the challenges comes from not being able to estimate the data properly. But now with sequencing data and ever larger data sets, the day where we can basically regress the trait on a lot of SNPs on the right hand side, at least like within a LD blot, estimates all the data is not that far, I think. Do you think that's correct? And do you think, there will still be G by E that differs by ancestry, but at least if you can control the nearby SNPs, you get much better estimates.
So that solves some of the LD problems, but that still doesn't solve the problems of differences in allele frequencies, right?
I fully agree.
Mm-hmm.
Well, it solves the allele. If you have a chip, if you have a sample that's just Europeans and it won't capture variation in SNPs that only vary among Africans, so that you won't be able to estimate the data for that. But for the SNPs that's only varying in Africans. But it will. So what do you mean by okay?
Yeah, I think you're right. So that would be a step in the right direction. In practice, so for example, in EA4 we try to predict education in an African ancestry sample, and if it were just the case that the attenuation was due to LD differences between European and African ancestry individuals, then we would be able to predict how much attenuation there should be, and we in fact see a lot more attenuation than we would expect if it was just being driven by LD. And so, there are other mechanisms that are causing problems for these types of analyses, right?
Well, but that's with genotype data where, the chip, okay, we can talk after.
Okay, yeah.